All right, now what I've done is I've added a bunch of questions that if you can answer, then you are doing great. So we'll go through these, and then you can also print this out and quiz yourself. All right, so when an atom is reduced, it gains or loses electrons. When an atom is reduced, it is gaining electrons. When an atom is oxidized, is it losing or gaining electrons? When an atom is oxidized, it is losing electrons. Is an oxidizing agent itself oxidized or reduced because it loses or gains or electrons or oxidizes or reduces a second atom? So an oxidizing agent itself is reduced because it takes electrons or it oxidizes a second atom. So an oxidizing agent will oxidize a second atom, and in the process of oxidizing a second atom, it will remove electrons from that second atom, which means the oxidizing agent is itself reduced. And reducing agent is itself oxidized because when a reducing agent reduces a second atom, it gives electrons to the second atom. In cellular respiration, glucose is oxidized or reduced. Glucose is oxidized during cellular respiration. A way to remember that also is glucose is broken apart to get the electrons out, which is oxidation. The 4 stages of cellular respiration are: the first stage is glycolysis, second is the prep step, third is citric acid cycle, fourth is oxidative phosphorylation. Number 7, which stages of cellular respiration take place in the cytoplasm? So that would just be 1, and that would be glycolysis. The prep step begins in begins in the cytoplasm. So part of the prep step takes place in the cytoplasm, and then the second part of the prep step takes place in the mitochondria. And which stages of cellular respiration take place in the mitochondria? That would be citric acid cycle and oxidative phosphorylation. Where does the citric acid cycle take place? Specifically, it takes place in the matrix inside of the mitochondria. Another name for the citric acid cycle, there's 2, the TCA cycle and the Krebs cycle. Where does the electron transport take place? So that's the inner membrane of the mitochondria, also the cristae. Where does active transport take place in the electron transport chain? Active transport takes place in the inner membrane of the mitochondria. Where does passive transport take place in the electron transport chain? That's a trick question because there is not any passive transport in the electron transport chain. That's all active transport. Where does active transport take place during chemiosmosis? That would take place in the inner membrane of the electron, of the inner membrane of the mitochondria. I'm sorry, no active transport takes place during chemiosmosis. That is all passive transport. So where does passive transport take place during chemiosmosis? It takes place in the inner membrane of the mitochondria. What are the two parts of ATP synthase? ATP synthase has one part transport protein and one part enzyme. The transport protein transports protons passively, and the enzyme phosphorylates ADP. Where in the mitochondria is the concentration of protons the highest? That would be in the inner membrane space. The inner membrane space has the concentration of protons increasing because the electron transport chain is pumping protons there. And where in the mitochondria is the concentration of protons the lowest? That would be in the matrix. And that's because the electron transport chain is actively pumping protons there. Question 19. Protons move by passive transport in a mitochondria from, so passive transport is from the inner membrane space to the matrix. And protons move by active transport from the matrix into the inner membrane space. Question 21. In what stages of cellular respiration is carbon dioxide released? So carbon dioxide is released during two of the stages. The first being the prep step, and the second being the Calvin cycle. In what stages of cellular respiration is ATP produced? There are three stages where ATP is produced. First is during glycolysis, and that is substrate level phosphorylation. Second, ATP is produced in the Calvin cycle. That is also substrate level phosphorylation. Then ATP is produced during oxidative phosphorylation. And that process is also called oxidative phosphorylation. And a majority of the ATP produced is during that last stage. In what stages of cellular respiration are electron carriers reduced? So when they're reduced, they are gaining electrons. So that would be in three of the stages: glycolysis, the prep step, and Calvin cycle. And technically, the electron carriers are oxidized during oxidative phosphorylation. Where do electron carriers deliver electrons during the cellular respiration? That would be the electron transport chain. And what happens to electron carriers when they drop off their cargo electrons? They are oxidized. Number 26, what is the final electron acceptor in cellular respiration? And that is oxygen. And where does the final electron acceptor accept the final electron? The oxygen is in the matrix when it accepts the final electrons. And then it makes water during that process. Which region of the matrix is more acidic? Which I should say which region of the mitochondria is more acidic, matrix or the inner membrane space. So a high proton concentration is more acidic, and the inner membrane space has the higher proton concentration, so therefore the inner membrane space is going to be more acidic than the matrix. 29, when is glucose completely oxidized in cellular respiration? During the citric acid cycle. So that would be in the matrix. When and where is water produced during cellular respiration? So there is some water produced during the citric acid cycle. And then water is produced in the matrix when oxygen accepts the electrons from the electron transport chain. What is the purpose of the electron carriers during cellular respiration? The purpose of the electron carriers are to take high energy electrons to the inner membrane of mitochondria in order to set up the proton gradient. So the electrons from the electron carriers are going to be used to actively pump protons into the inner membrane space from the matrix. So the first immediate purpose of the electron carriers is to deliver electrons to the electron transport chain. That would be the first immediate job. And then those electrons are used to actively pump protons from the matrix into the inner membrane space. And the purpose of the electron transport chain is to make the proton gradient that will power ATP synthase. What would happen if the electron transport chain became non-functional? So the first immediate consequence of the electron transport chain being non-functional, so if it stopped working, the first immediate response would be that protons are not being pumped from the matrix to the inner membrane space. And then a second and third consequence, so what would happen once the protons are not being pumped across the inner membrane? The proton gradient would eventually run out because the protons would then just keep moving by diffusion back into the matrix. So the second consequence would be that the proton gradient would start to decrease. So it would be moving towards being isotonic. And if we don't have the proton gradient, then ATP synthase is not functioning. So we would stop making ATP. So these types of questions where, something's gone wrong, something doesn't work anymore, you always want to think of the very, very first immediate consequence. And then what would be the second consequence? What's the third consequence? An incorrect answer would be cellular respiration would stop. No duh, of course it would, but that's not really the question. The question wants to know what is the first immediate consequence. What stage of cellular respiration is also a part of fermentation? That is glycolysis. The two types of fermentation, how are they similar? How are they different? Where does it take place? And does it require mitochondria? So the two types we learned about are lactic acid fermentation and alcohol fermentation. They're similar because they both include glycolysis. They both take place in the cytoplasm. they don't require oxygen. They both produce NADH, so they both reduce NAD+. They both make ATP, 2 molecules net of ATP. They're different because they have different end products. Lactic acid fermentation, the product is lactic acid. Alcohol fermentation, the final product is alcohol. And then also alcohol fermentation produces carbon dioxide, and lactic acid fermentation does not. They don't require mitochondria at all. So prokaryotes can use fermentation. Why is the inner membrane of the mitochondria highly folded? So it's highly folded because that increases the surface area of the inner membrane without making the mitochondria humongous. So we learned in, I think, our second unit why cells are small. One way cells can increase the surface area of their membranes is by all of these folds, folding over on themselves, etc. That's a way to increase the membrane, but keep the overall cell small or organelle. Okay, then at the end here, I gave you some examples of how to look at material to try to predict application questions. So, for example, glucose is oxidized to 6 molecules of CO2. So that's really, really important part of cellular respiration is the oxidation of glucose into CO2. So something you would want to keep track of would be all 6 molecules of CO2. Like there can be an application question in there somewhere because you could be asked a question and then you only end up with 5 molecules of CO2, or you end up with 8 molecules of CO2. These kinds of correlations, you want to watch for when they aren't what you expected. Because then that means, okay, if you've got too much or too little, what would cause that? And the way that this question would be asked, the question would not point out that you either had too few or too many molecules of CO2. That would be up to you to catch that. So that is asking you, do you realize that there has to be 6 molecules of CO2 produced for every glucose molecule? So that could be a very subtle application question. So taking a fact from the material and having it presented to you where the fact is not correct, but it should be. So you've got to figure out where something went wrong. How could you get unexpected data? What would cause unexpected results? And then also, we have a solid that is being converted into a gas. So that's would be visually obvious. You've got this gas being produced from a solute, and then you're also in an aqueous environment. So something like that, that's visual, or you could see it happen, or you could what would be the consequence of this gas like not getting out of the cell? So these changes in states of matter. Anytime a gas is produced, you should always, in general, it's a good way to live your life. Just kidding. But if a gas is produced, you really should take note of that, because that could be an application question. All right. The fact that cellular respiration takes place in 2 locations in the cell, that should raise a red flag. And you should be mindful of the questions and how they or what they are asking you about location. That could be easily an application question, maybe a molecule not in the location you would expect, or a different molecule in that location. Something else has gone wrong, and now there's a molecule there that wasn't before. How is that going to affect your molecules that are participating in cellular respiration? So if there's a change of location, then keep an extra eye on the question to see if they throw a wrench in there and the molecules are not in the correct location. The concentration gradient, the direction of the concentration gradient. When you've got a collection, whenever you've got a concentration gradient at all, actually, take extra time and look to see that you understand what is expected, and then what would unexpected look like? What could cause unexpected? There's a lot of application questions possible in any kind of concentration gradient because the concentration gradient drives movement. So if the concentration gradient is gone, then some molecules aren't moving anymore. And then what's the second consequence? And what's the third consequence? So you get this chain of reaction of unexpected events. So concentration gradients should always raise a red flag because there are going to be more processes dependent on the concentration gradient besides just that one molecule that's moving by diffusion. Now, and the proton concentration, any kind of proton concentration, you should immediately think pH changes. Is this region going to be acidic? Is it going to be basic? And if it becomes too acidic, what's the consequence of a region becoming too acidic? And then in general, with any kind of reaction, you need to be aware of products piling up or reactants piling up, byproducts piling up, these unexpected results. Maybe there is one of the reactants is getting stuck somewhere because a proton pump or a transport protein is non-functional. What happens if your reactant's concentration is decreasing? What if your reactants cannot participate in the reaction? What happens when the reactants are just sitting around? Are they really, really reactive? Are they going to shut other processes down? So these are things that you should look for when trying to anticipate application questions. In general, thinking about what can go wrong, what can pile up, what can be distorted. Anything with a double membrane, you always get questions where, something's happened and the membrane's been disrupted and it, breaks off and forms a new organelle. Which way is ATP synthase pointing? Where are the protons moving? Etc. So understand the material so that if there are changes made, if a new protein is embedded in the membrane, etc., etc. So understand the facts and understand the process that goes with the facts. All right, let me know if you have any questions.